Okay guys, so we're gonna do our continuation from our disinfectant lab we did previously. So we did our other lab, we checked the effectiveness of disinfectants on E. coli or Staph aureus. Now I'm gonna do the same thing with antibiotic discs. So this is gonna simulate the Kirkby-Bauer test. So you'll also be checking for zone of inhibition using antimicrobials, all right? So if you look here, I've got penicillin, erythromycin, and triple sulfur. And I've written down here, for penicillin, you guys remember, this is a cell wall inhibitor. Usually works on gram-positive bacteria. We know we have some that are penicillin ACE producers, which are penicillin resistant. And we have MRSA, which is methicillin resistant, Staph aureus, and so on. So we're going to just test the basic effectiveness of penicillin. Erythromycin from lecture and lab, we said that's a ribosomal inhibitor. I'm not going to ask you guys if it's a 30S or 50S inhibitor. It doesn't matter at this point. And our last one is our triple sulfur, which inhibits folic acid. And we discussed in lecture and lab that we need DNA. We need folic acid for DNA production in the humans. Bacteria can produce it. We can't. That's why it's so important that you guys, females of childbearing years, take our folic acid, B9, so we don't have any birth defects in that first trimester. But anyway, that's a side note. So what I'm going to do today is I'm going to swab the plate like I did with the disinfectant. So I'm going to swab one with E. coli, which is our gram-negative, and one with Staph aureus, which is our gram-positive. All right. So let me just grab a swab here, sterile. All right. And I'm going to grab some E. coli off of here. We've got some colonization here, not a whole lot, but enough so we can lawn this. All right. So I'm going to lawn this. Make sure you don't cross the line. All right. So we'll just check 3 antimicrobials again. Once again, penicillin, erythromycin, and triple sulfur. All right. So I'm going to lawn the Staph aureus. It's going to be our gram-positive example. All right, and once again, like I mentioned with the disinfectants, the gram-negative should be slightly more difficult to kill. It has those 2 cell walls. Right. So I'm going to try not to drop these. Right. So where is it? It says penicillin right there, if you can see that. Right. So sterilize this. All right. Pop that disc out there. Pop another one out here. So once again, we have one for our gram-negative and one for our gram-positive. Okay. Well, that's no longer sterile, but it's all right. So this is going to be our number 1. Good Lord. Now normally whenever you drop these you just leave them here, but that was too close to the side. So we want to make sure we can really check our zone of inhibition. That one. Wow, that one way better. All right. So we have one in our E. coli, one in our Staph aureus. All right, let's move on to our erythromycin. Remember, once again, this is our ribosomal inhibitor. Right. So we know that all bacteria has ribosomes. All right, so this should definitely work. All right. And if you guys remember, the only problem with these are that mitochondrial RNA and bacterial RNA are the same. So the only issue with these sometimes is that some of these can affect human ribosomal mitochondrial. So it says E for erythromycin. This one says P for penicillin. We're not going to worry about the dosage right now, because we're not weighing the patient. We don't know the age of the patient. Doesn't matter. Oh. Sulfur. Once again, this is our folic acid inhibitor. So unless they can find another pathway around the PABA, this should work. I told you guys before, bacteria are super smart, smarter than we are. They've been around a lot longer than we have. So a lot of things have figured out a way around this, but hopefully this E. coli and this Staph aureus is not. And we've got that in there. And once again, we want to make sure these are in there seated, but like I told you guys, don't Wolverine these. They don't have to be jammed in there. Right. And we will check the effectiveness of this on Monday, and I will post the results in lecture and lab, and we'll make a little video showing the results. We can measure the zone of inhibition, and we'll see how we made out. All right.